Dear viewers, I am Dr. Amit Bhattacharya from Ramjas College, University of Delhi. So we come into the second part of the lecture, which is basically on the history of modern biotechnology techniques and its application. So in the first part we discussed about what exactly the biotechnology is and what are its uses. Now in this part of the lecture I will be talking about the emergence of the recombinant DNA technology and the various processes after the 1970s. Now, the emergence of the recombinant DNA technology, it started in the 1970s. So, in the 1973, two of the scientists, Boyer and Cohen, they for the first time give the technique what exactly the recombinant DNA technology, because one of them were working on the restriction enzymes while the other was working on the systems in which the foreign DNA can be replicated and can be transformed into them. Now this particular image, if we see over here, is the basic conceptualization of the recombinant DNA technology. So we see that there is a plasmid cloning vector which is there. So the plasmid cloning vector, in the plasmid cloning vector, the insert DNA is incorporated or the foreign DNA is incorporated. Now with the help of the restriction endonucleases, this particular foreign DNA is incorporated over there. Now as it is incorporated, we form a recombinant DNA over there. Now this recombinant DNA contains the cloning vector as well as the insert DNA which has been inserted into the multiple cloning site within the cloning vector. Now then it is transformed into the bacterial system where it start growing up with the bacterial system. Now as this particular growth takes place, what happens is the gene which is present within these particular insert, they are also transcribed and translated. Now as these genes are transcribed and translated, the proteins are synthesized from them. Now this led to the foundation for the development of the first human insulin also. Now for this work, Boyer and Swanson and Cohen, they were recognized at the various platforms in the year 1976, and the subsequent development of the recombinant DNA technology gave way to the human insulin also. Now this is a basic structure how exactly the human insulins were produced in the bacterial host system. Now if we see over here, one of the plasmid vector, the insulin gene A was inserted, while in the other cloning vector system the insulin B gene system was incorporated. Now as this particular cloning vector was inserted into the competent cell or the bacterial cell, they start growing over there and they start transcribing and translating over there to produce the proteins over there. Now in the culture cells, as these proteins were produced, after some time they were isolated from these bacterial systems and then they were purified and classified. Now they synthesized the gene A protein as the gene B protein over there. Now we know that the human insulin contains the chain A and chain B which are attached to one another with the help of the disulfide bond, and subsequently with the help of the disulfide bond the exact structure of these human insulin was formed over there, and that's how the human insulin came into the picture. Now this formed the foundation for the development of the human insulin, and subsequently the various prototypes were produced, like the human growth hormone, somatostatin hormone, and the various growth hormones came into the picture. Now in the year 1975 to 81, there were various blotting techniques which also came into the picture, like the southern blotting, northern blotting, and the western blotting. Now the southern blotting technique was basically to detect the specific sequences of the DNA fragment with the help of the gel electrophoresis. Now in the northern body blotting technique, the specific RNAs were separated out and then they were detected with the help of the agarose gel over there. And the third one is the western blotting technique, which helped the scientists to identify the proteins which were present in the samples over there. Now these three blotting techniques also played a very important role in the biotechnology field. Now we come into the picture over here, which more or less show what exactly happens in these blotting techniques. Now first what happens is a gel is casted. Onto the gel there are various wells which are prepared. To these wells the samples are loaded, which can be a DNA, RNA or a protein sample which is loaded. Then the electrodes are switched on, so based upon the charge they are separated over there, and we know that the gels are having the molecular sieves, or there are small pores which is there. So the smaller fragment, it moves farthest in the gel, while the larger one remains on the top. So based on that, the separation takes place, and subsequently what happens is the gel image is then transferred onto the nitrocellulose membrane, where the fragments are transferred over there with the help of the various blotting techniques. And finally, the nitrocellulose membrane, it has got a replicate image of what have been separated in the gel. Now these nitrocellulose membrane are then added with the help of the various probes. So these probes are specific to the region which is of our interest. So these probes go and bind to the specific region within these membranes over there, and as the binding takes place, what happens is we can trace out or see the particular band which is glowing over there. So we can see over there is an autoradiography image which is showing over here. So the radioactive probe have been used, and this shows that this particular DNA segment which is of our interest is present within those particular samples over there. So with the help of these particular technique, we can detect the DNA of a given sequence. So this is a southern blotting technique which is shown over here, and the DNA segment, as well as with the help of the ladder which is run along with the samples over there, we can classify or give a comment what exactly can be the size of these particular DNA fragments over there. Now the next one comes is the northern blotting technique, which detects the specific mRNAs over there. Now the northern blotting techniques, it specifically detects the specific mRNA. What happens is first the RNA samples are isolated from the cells, then they are processed through various processing mechanisms, and finally they are run onto the gel. Now once the running onto the gel happens, they are again transferred to the nitrocellulose membrane by the blotting techniques which we just saw in the last slide. And once the transfer takes place, then the probes are used. The labeled probes, they go and specifically bind to the specific segments of these mRNAs, and they start giving up the colored signals over there. So there is an autoradiography image which is shown over here. We can see the specific mRNA samples are giving a glow. So we can easily say that this particular sample contains the mRNA of our interest which we wanted to detect. The last blotting technique is called as the Western blotting technique, which detects the specific proteins within the sample. So again, this is called many times referred as the polyacrylamide gel electrophoresis. So the gel is prepared, onto the gel the samples are loaded. So again the samples are separated based on their sizes. So the smaller one travels faster while the larger one remains on the top, and once the separation takes place, what happens is the probes are added. They are transferred onto the nitrocellulose membrane, then the probes are added. The labeled probes, what it does is it go and binds to the proteins. So basically in the case of the proteins, the probes are basically the primary antibodies which can detect the specific protein or the antigens present within the proteins. So they will go and bind to these proteins over there, and once the binding takes place, they contain specific radioactive part region over there which gives a specific signal over there. So in the autoradiography image, we can clearly see the banding pattern. So if we see the bottom image over here, we can see a ladder has been run, as well as in the various samples the protein has been detected over there. So we can see a clear black spot in that particular sample, which gives us an image that that particular protein is present within that particular sample or not. So they help us to detect the specific proteins over there. Now again, this particular technique is many times referred as the PAGE, which is the polyacrylamide gel electrophoresis. So acrylamide gels are used over here. So the selective size are being separated. So as this protein moves through the gel in the presence of the electric field, the smaller molecules, they travel faster or they rapidly than the larger molecule. So there is a separation of these protein molecules which takes place, and then subsequently they are transferred onto the nitrocellulose membrane, and then they are probed. With the help of the probe, the specific DNA, the specific protein molecules are being recognized over there. The next important discovery which took place in the year 1977, and this was basically the sequencing of the DNA. So one of the scientists, Sanger, he first came up with a technique which was called as the Sanger sequencing technique. Apart from that, there were two other scientists, Maxim Gilbert, who also came with the chemical modification sequencing DNA technique over there. Now, for this particular work, Sanger got a Nobel Prize in the year 1980 for the sequencing out. Now, what the scientists did, very interesting, is they used the concept of the DNA replication over here. Now, we know that in the DNA replication, one after another, the nucleotide based on their base pairing, they come over here. Now, with the help of the phosphate groups over there, the phosphodiester bond is formed between the incoming and already present nucleotides, and subsequently the chain is elongated. So if we see the animation image over here, this animation image shows how the DNA replication is being done over there. So we can see over there the A corresponds to T, G then binds with the C nucleotide over there. So that's how the replication start taking place. Now they use this particular concept to form and modify this technique. Now what they did is, Sanger, what he did in his technique is, he used, instead of the dideoxyribonucleic acid, the DDNTPs over there. Now if we see over here, what happens is, in the dideoxyribonucleic acid, there is a OH which is present in the third carbon, but in the presence of DDNTPs, the OH in the third carbon is missing. So as soon as these nucleotides were added over there, what will happen is the elongation will stop taking place over there, because there won't be any formation of the phosphodiester bond with the incoming nucleotide over there. So the break will take place. So this concept was used to form the basic conceptualization of the DNA sequencing, which was called as the Sanger DNA sequencing technique. Now this is a basic image how exactly the Sanger sequencing was done. Now if we see over here, there was a DNA template which was there, and there were 4 reaction mixtures which were there. So in one reaction mixture, apart from the DNTPs, the polymerase enzyme, the primers and the buffers, the DDATP was added. In the second one, there was a TTP was added, third one DDCTP, and the last one was DDGTP, which was added over there. So as the DDNTPs, they were coming up in those particular reaction process, they were blocking or stopping the reaction or the sequencing process at that particular moment, due to which there will be fragments which will be produced of various lengths over there based on the addition of that particular DDNTP over there. Now this led to the foundation of understanding the sequencing process. So all the samples from the 4 reaction mixtures, they were put onto the well and then they were run onto the gel. So based on that, there was certain bands which were produced over there. So now if we start reading the bands from the bottom to the top, we get a sequence of the DNA which will be present over there. So based on these particular things, they discovered that this particular method or the technique can be utilized in the discovery or sequencing out the DNA techniques over there. Now this was called as the technique which was called as the Sanger sequencing technique. Now the next important discovery was taking place in the year 1984. This was called as the DNA fingerprinting technique, or many times referred as the DNA typing over there. Now this particular technique was developed by the British geneticist Alec Jaffray. So what he noticed is, there is a unique segment of DNA which is present within each of the person, which is unique to an organism. These were called as the mini satellites. So these mini satellites were present within the genes of a DNA sample, and they varied in their numbers actually. Now we will see what exactly is he wanted to suggest. Now if we see, this is a human chromosome which has been shown over here. In a particular segment of that human chromosomes, it has been magnified over there. Now this particular segment has got various restriction endonucleases or the cutting sites over there. Now we'll focus into 2 two of the cutting site. Within them there is a VNTR which is present, which is called as the variable number tandem repeats over there. Now this particular number, they vary among the individuals. What happens is, if we see in the first case, there are about 14 variable number tandem repeats which are present. In the second one there is about 9, and the last one there are 4 variable number tandem repeats which are called as the VNTRs. So the VNTR regions are polymorphic in nature, which means that each person is having a specific number of these particular VNTR regions within them. So all the population individuals can be identified and classified based on these VNTR regions. So this is the basic technique which he used over there, and now it is being used into the forensic experiments and the forensic sciences. Now what he did is, he said that suppose from the forensic sample region a specific sample is being collected of the person who has done that particular crime activity, then the suspected individual's DNA samples are collected. Then the DNA samples are cut with the help of the specific restriction endonucleases, and then they are run onto the gel. Apart from running the gel of samples of individual A, B and C, the samples which was collected from the forensic side, it was also run onto the gel. We know that each of the individual are having a specific and a different variable number of the tandem repeats within them. So all the persons will be different in that particular segmentation part. So based on these particular DNA fragments, there will be various fragments which will be produced in sample A, B and C, and suppose if one of the samples, it corresponds, it looks similar to the forensic sample which was collected from that particular area, it gives a clue that that particular person is the one who was involved in the crime region over there. So if we see over here, the DNA fragment pattern in the case of the individual B is similar to what has been found from the forensic sample, and we can easily confirm that yes, the individual B is the one who is involved in that particular crime over there. Now the next important discovery was what happened in the year 1987, for which the Carey Mullis got a Nobel Prize in the year 1993, and this was basically the polymerase chain reaction. Now polymerase chain reaction, as the name is suggesting, what is happening is the polymerase, the enzyme, what it doing is, it is synthesizing a number or replicating a number of copies of a small DNA fragment. So whenever we have got a small DNA fragment in a very low titer concentration, what we can do is we can easily run a polymerase chain reaction to increase the number of these particular proteins. So this was another important discovery which was done in the field of the biotechnology. Now again, this particular technique used the technique of the DNA replication. So in the DNA replication, we know that we require the DNA template strand, which is the single stranded DNA, the primer, which are both forward and reverse primer, the DNA polymerase, the dNTPs, which can be the dATP, dCTP, dGTP and dTTP. So one after another the incoming nucleotide comes, and based on the base pairing, they come and bind over there and form the phosphodiester bond over there. Now once the phosphodiester bond has been formed, they subsequently form the daughter strand over there on the top of the DNA template strand. So this particular mechanism was again utilized in the conceptualization of the DNA, the polymerase chain reaction or the PCR mechanism. Now what he did is, the polymerase chain reaction, there were three major steps which were there. First was the denaturation, annealing and the elongation step which was there. The denaturation takes place for 1 minute at the temperature of 94 degrees Celsius. So what happens is, the DNA double helix structure, it gets denatured, so the base pairing gets ruled out from there. So both these strands get separated. So now we have got single stranded DNA which is there, which can form the template strand. Then the annealing takes place, means the primer gets bound. So the forward and the reverse primer binds to both the strands over there, and once the primer has been attached at a temperature of 54 degrees Celsius, the elongation takes place, which takes place at the temperature of 72 degrees Celsius. So in the elongation step, the DNTPs, which can be ATP, GTP, CTP and TTP, they are added. Apart from that, there is a Taq polymerase, the enzyme which helps in this synthesis process, also start taking place. Now the Taq polymerase enzyme is a very interesting enzyme because it has been isolated from a bacterial system which is called as the Thermophilus aquaticus, and it can sustain a higher temperature. So because in this particular steps there were increase and decrease of the temperature, so the polymerase enzyme have to be very stable at the high and the lower temperatures also. So the Taq polymerase was utilized over there, and once the elongation takes place, what happens is the template strand is elongated, and once the elongation takes place, the daughter strand is formed and the DNA double helix structure is formed based on the base pairing rule. So this was called as the PCR technique, which is called as the polymerase chain reaction. So if we see over here, this animation image gives a basic overview what exactly happens in the PCR technique. So first we saw that there is a denaturation step, so we can see the DNA double helix structure have been separated. Then the annealing step takes place where the binding of the primer takes place, and finally the extension takes place at 72 degrees Celsius, where the extension of these primer takes place with the help of the base pairing rule, and the daughter segment or the strand is formed. So now again the DNA forms into a double helix structure, and from one copy the two copies are formed, from the 2 to 4, 4 to 8, like this one, how the multiplication start taking place. So the PCR technique also played a very important mechanism in the biotechnology inventions. Now the next important discovery was a number of vaccines which were discovered with the help of these recombinant DNA technologies. One of them is called as the vaccines for the hepatitis B virus over here. Now in the hepatitis B virus, we know that as soon as the virus infects the person's immune system, what happens is the immune systems get activated, especially the plasma cells get activated. Once the plasma cells are activated, the antibodies which are specific to these particular viruses are secreted within the body over there, so that these antibodies will go and neutralize the viruses which are circulating within the body systems. Now this particular mechanism was used, and what the scientists did is, because they knew that the antibody which is present within the circulation, they can recognize the specific antigen or the specific sites within the surface of that particular virus. So these are called as the surface antigens. So what the scientists did is, they came up with a recombinant vax HB system in which the hepatitis B vaccine was developed over there. What they did is, they first isolated a surface antigen which was called as the hepatitis B surface antigen, which was isolated. Then this particular insert was inserted into the bacterial system, and it was transformed into the competent cells, where it replicated over there, produced a number of copy of these particular segments, and finally they were purified and bottled into the vaccines, which when incorporated or injected into a healthy person will elicit a immune response or the B cells or the memory cells for subsequent infections within that particular individuals. So this particular led to the foundation for the vaccines technology also. Now another important technique which came into the picture was in the year 1995, which was called as the microarray technique, in which the scientist Mark Skenna, what he did is he took out all the genes or cloned each of the genes of the whole genome of a person or an organism onto a small surface or a special chip. So what he did very interestingly is, onto a chip all the genes were cloned and they were attached over there. So these, if we see the chip over there, they were attached with the DNA over there, and these DNA are the single stranded DNA which is there. Now the samples from the persons is taken up. So suppose a person is suffering from a disease condition, the samples are taken up, they are probed, labeled samples which are done, which can be fluorescence dye in this particular case, and once the fluorescence dye labeled are there, they are attached over there. So the probe DNA is attached over there, and once the samples are attached to these particular thing, they will attach to the specific region within them, and whenever that particular region is being scanned with the help of the various detectors, the hybridized probes will give a specific coloration or specific signal over there. So the scientists can easily make out that these particular gene are being activated, are being present in a higher copy number. So these particular technique came into the picture, and all these things came into a small miniaturization was done onto a small slide over there. So it helps the scientists to simultaneously evaluate the gene expression. So in a single slide, all the genes were incorporated, probe DNA was incorporated, and the person's samples were added to it, and the scientists can easily make out which are the genes which are upregulated, which are downregulated, or which are present in a normal conditions over there. Now, if we see over here, this is a 2-color fluorescence microarray probe which is shown over here. So we can see over here the normal cells which are shown in green color and the cancer cells which are shown in red color. So as the samples were isolated and then they were probed, they were hybridized onto these chips. What happened is the red color signals were basically the upregulated genes. So this gives a scientist idea what are the genes which have been upregulated in the case of the cancer cells, and the green color genes are basically the downregulated gene, while the yellow one are the normally expressed gene. These microarray found the foundation for the subsequent techniques which got into the picture. Now the next important assays which came into the picture based on these particular hybridization techniques was called as the fluorescence in situ hybridization, and many a times it is referred as the FISH technique. So the FISH basically employs the fluorescent probe to detect a specific DNA sequence in the chromosome. So the FISH technique helped the scientists to scan the various chromosomes and various parts of these things. Now the FISH technique were published in the year of over there, and the various forms of these FISH techniques help the scientists to go into the various forms of the cytogenetics. So with the help of these cytogenetic techniques, they were able to detect the specific gene within that particular person's sample, and they can classify whether that particular person is sampling or suffering from that particular disease condition or not. So with this note, I come to the end of my lecture. So in this part of the lecture, we took up some of the modern biotechnology techniques which were discovered, and we discussed about the various techniques, and in the next part of the lecture we will discuss further more of the biotechnology techniques which were discovered subsequently after 1980s part, and how they are helping in the diagnostic of the various disease condition, the medical field, the agro field, and the agricultural field, development of the genetic modified plants, animals, and these particular things. So with this note, I come to the end of this first part of the lecture. Thank you very much for listening. Thank you.